Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Epen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@epen.info. Hi, everyone. People familiar with Scientific Sense podcast know that we typically have one guest, and today's session is a little different. We have three other guests, and we're going to have a broad conversation around microbes, the microbiome, how microbes interact with other biological systems, not just humans. And so we have three guests, and I'll let them introduce themselves. Fred, do you want to go first?
Sure. Well, thanks for having me on, Gil. My name is Fredrik Inglis. I'm an assistant professor at the University of Missouri in St. Louis. And I study microbiology. I'm particularly interested in how microbes evolve and interact with each other and how these interactions cause disease.
Brittany?
Hi, thanks for having us, Gil. I'm Britt Peterson. I'm an assistant professor at Southern Illinois University, Edwardsville, and I study insect microbe interactions. We colloquially refer to my lab as the bug guts lab, and we're interested in how complex microbial communities influence each other and their hosts to manifest as traits that are selectable and how they evolve as a collective, basically.
Sussane?
Hi, thanks, Gil. I'm Sussane DiSalvo. I'm also at Southern Illinois University, Edwardsville, so SIUE. I'm an assistant professor, and I study the interactions between bacteria and eukaryotes, specifically bacteria interactions in a sort of model microbial eukaryotic system to look at the mechanisms and outcomes of those interactions, specifically intracellular bacterial infections in that system.
Excellent. So before we start, I want to set the context a little bit, and I want to go all the way back. So the universe started about 14 billion years ago, the solar system and the Earth around 4.5 billion years ago. My understanding is that the first microbe showed up on Earth about 4 billion years ago. And so they have been around for a long time. One could argue the bacteria, probably the most successful life that we know of. Definitely on Earth, and we haven't found anything else outside the Earth. So they still seem to be one of the best biological system that we know. So 4 billion years is a long time. So I would imagine they have evolved, and they evolved with other systems, maybe at least part of that 4 billion years. So what do we know about sort of the evolution of microbes?
Yeah, so I mean, you raise an interesting point there, Gil. And I think some of the things to keep in mind, I think it's quite hard for us as humans. We're very visually dominated. We tend to be interested in things we see, but really we live in a microbial world. The vast majority of life on our planet is, are bacteria or prokaryotes or even eukaryotic microbes, just tiny little things that we'll never be able to see with the naked eye. And they in fact dominate all of the important geochemical processes, from nitrogen fixation, carbon cycle. These are mostly microbial processes. It's true that some big things like plants get involved occasionally, but really it's microbes that run the show. And they've been running the show for about 4 billion years. And then at some point, eukaryotes evolved, and multicellular eukaryotes evolved, that, things like humans. And even in terms of like human life or other kind of insect life, we wouldn't be here if it wasn't for the microbes inside of us and on us. And so I think this is a kind of we have this growing appreciation that many of the processes, even in humans, require microbial life. I think one of the kind of classic things you'll hear people say is that there are more bacterial cells in the human body than human cells. And that's in part because they're so tiny. That's also incorrect. There's probably about an equal amount of bacterial cells as are, human cells. But that's a huge number. There's probably like, a few pounds of your weight is bacteria in your gut. So, and if you don't have bacteria, like if you're not a biotic or like you get rid of all the bacteria, you don't live very long. And so they're essentially very important for all kinds of processes. And we're I think, it's an exciting and growing field trying to understand what these bacteria are, what they're doing, and then how we can even manipulate them to try to, cure diseases or prevent kind of infections.
Yeah.
Yeah, and I think one thing is, if you think about our own evolution as just eukaryotes, it's required that, or the idea is that endosymbiosis basically initiated the occurrence of the common eukaryote now. So a endosymbiosis event wherein a ancestor of the eukaryote basically engulfed a bacterial cell, and then that bacterial cell evolved into our mitochondria and possibly other organelles. So this event probably occurred a few times, depending on the particular eukaryote you're talking about. And that allowed for this evolution of eukaryotes to begin with, and then of course the evolution of multicellularity.
Yeah, and I really think that, kind of going back to what Fred was talking about, a lot of the physiological and even ecological processes that we kind of attribute to even large eukaryotes like plants, they have a microbial component. And if we lived in a germ-free world, those things would cease to exist. And so I think the impact of microbial life can't be overstated. It really, they seem to be the puppet masters in almost all systems when it comes down to it. We don't understand all the mechanisms and the underpinnings, but we do know that when you take them away, a lot changes.
Yeah, we will talk more about this, and I think this idea is getting a little bit more debated, but some people say that the human body is really an enclosure for the bacteria to make a living. But I think there's more debate around that. So, Britt, one of your area of interest is looking at not just human guts, but other pests and other things, right? So how bacteria might affect them, and just like humans require certain type of bacteria in the gut to process food and so on, we see that it's got a sort of a common problem for other organisms too, right?
Yeah, I think this this pattern of an animal being colonized by some number of microbes is pretty typical. Who those microbes are and what they're doing is variable. And how obligate that interaction is also variable. But it seems to hold true that there are microbes in these systems, and we can understand various things about the host based on studying those microbes. So the systems that I study, like termites, we've known for 100 years that termite digestion of wood is dependent, at least largely, on a healthy gut microbiota. But we're learning more and more that, their immune system is also really primed by the community in their guts. And we're learning more about how gut microbiota influence things like circulating hormones, and that can have implications for behavior. And so it seems like their little flagella are really kind of getting into just about everything we identify as being host related. And so while the cast of characters is different and the manifestation of that cast is different in each host, there seems to be some microbial influence.
Yeah. And Sussane, you look at sort of the interaction between bacteria and other microsystems like amoeba. So to set the sort of the size of these things, so am I right in thinking about bacterium at about 1 micrometer and maybe an amoeba maybe 10 times the size of that?
Yeah, that's about right.
Okay. And so what do we know? I know that you run a lot of experiments around this. So what do we know about the interactions at that scale between bacteria and amoeba in this case?
So in this case, the amoeba that I look at is a soil-dwelling amoeba, and so it actually is constantly consuming bacteria as a food source, and it phagocytoses those bacteria and digests them. And what's interesting, I think, about that too, if you want to relate that to, say, a human system, is that this is similar to the way that some of our innate immune cells function to actually clear out bacteria and other sort of microbial infections from our body. So they actually phagocytose those pathogens and digest them. And that's actually what these amoebas are doing in the soil naturally to acquire food source. And then, of course, in the soil, there's just so many of these microbes that are interacting there. And so you have these dynamic interactions from, say, the predatorization pressure that the amoebas are inducing on the bacteria. It's also bacteria potentially evolving strategies to evade that, or even to establish as sort of intracellular parasites within the amoebas. And all of those mechanisms may also be similar to, say, the interactions that bacteria may have for higher organisms like mammals, and could even lead to the evolution of, say, bacterial pathogens for humans.
And so amoeba is using at least some variety of bacteria as food.
Yeah.
So they're basically processing the bacteria and converting that into food. So they're not really staying within the system.
Right. Yeah. So for the most part, they're pretty capable of consuming, it seems like, a wide variety of bacteria as a food source. And some bacteria are able to evade that, and some may be able to, be intracellular parasites. But from, I think, the larger scale, it seems like these amoebas are pretty efficient at predatorizing a huge variety of bacteria. Not just bacteria, it's been shown that they can even eat other larger microbes, such as fungal cells, which they're about equivalent in size to.
I think also another interesting point about these amoebas, not that they only eat bacteria, but they might form sort of symbiotic relationships with some bacteria. And this is controversial. So Sussane may contradict me on this, and that's fine as well. But there is some evidence to suggest that some types of bacteria can confirm some type of benefit to these amoeba. They can either detoxify the environment, prevent perhaps infections by more pathogenic bacteria. How stable these relationships are, or how transitory, we don't know. It might just be that it happens occasionally. It may not be dominating these interactions. But I mean, it is tantalizing to think that even in amoeba can hold tens, maybe 100, well, that's probably a lot, tens of these bacteria, and these bacteria might actually be forming some kind of symbiotic relationship or beneficial relationship between both the amoeba and bacteria. It's
right, yeah. Yeah, and I don't think we really fully know the ecological relevance of that, or as Fred said, even the amount that this is occurring in the environment, or how stably it's occurring in the environment. But the other component of it is it could be very context dependent, whether these types of interactions could be beneficial to the amoeba or harmful to the amoeba. So I think all of that we really don't know, and that probably expands to pretty much every many of the interactions that are occurring in nature.
I know it's certainly true in insects. Context dependence is everything, and for a lot of these interactions.
Yeah. So talk about that a little bit, Britt. So detoxification. I want to sort of explore detoxification aspects, but also, I don't know if this is practical, but are there any therapeutic effects of this bacteria eating amoeba on human systems?
So I can speak to the detox question.
Yeah.
So we know, at least in insects, that these interactions with bacteria can be relatively transient, but confer huge benefits to their host. So there's examples in some insects where in a single generation, a population of bugs can pick up soil bacteria just, in their normal being bugs. And those bacteria, because they've been exposed to particular pesticides for, there's some selective pressure there, they're able to break down those pesticides and use them as things like carbon and nitrogen sources. And in insect hosts, they continue to do this kind of basic metabolism, and it ends up benefiting the host. And so having some kind of gut lining or particular symbiont organ that can be colonized by these bacteria can confer massive advantages and can sweep through a population really quickly. And it's as simple as these bacteria do it normally. It's like bioremediation, but in an insect. These bacteria do it normally, and these insects are basically just biotech, right? They're exploiting these bacteria for what they do normally, and it confers a benefit.
So the pesticide resistance that we are seeing, part of that is related to this effect that the bacteria are really helping this pest to get around the pesticides?
It can be. It's definitely not the only way. There's selective pressure on the host to detoxify or mitigate it as well. Especially in some of our American agriculture systems, there's a huge selective pressure on the host as well. But the bacteria are in the soil and on plants or in the host themselves, so they're facing the same pressure, and it's an abundant nutrient. If you can think about a pesticide as a potential nutrient, it has carbon, sometimes nitrogen or phosphorus side groups. That's to a bacterium, that's food. You just have to have the right enzymes.
Yeah, so this goes into a broader question of the symbiotic relationship. And so if you look at it as a system, the pest and the bacteria inside the pest, that relationship ultimately is going to counteract the pesticide. So from an evolutionary perspective, can we think of that system evolving, or it's not that simple?
Sometimes. What's really interesting is a lot of these interactions seem to be pretty facultative, and they will pop up and then just as quickly they'll go away. A lot of gut microbiota are not intracellular. In contrast to some of the things that Sussane was talking about, they're actually extracellular. They're colonizing the walls of the gut. And so there's less reliance on the part of the bacteria for that host. If the pesticide is in the soil and the pesticide is in the host, they don't necessarily need to be in the host. It confers a huge advantage to the host, of course, but the bacteria may or may not have a selective advantage being inside the host. So we have examples where there seems to be a really tight coevolution of a host and a bacterium. And then we have other examples where it seems to be pretty transient and they pop kind of in and out, and it's population dependent and context dependent.
I think so. Just fascinatingly, riffing on what Britt was saying there are some insect examples where maybe we're seeing this kind of where we can actually start thinking them as a system that maybe evolution is acting on. There's this kind of alignment of interest, a bit almost like our mitochondria inside human cells. They're part of our cells, but they have some origin that's outside, some bacterial origin from billions of years ago. And so one of these examples is in cicadas. They have these bacteria called Tremblaya, and there's a number of other ones as well, but they have tiny, tiny genomes. In fact, they can't survive outside of the cicada host, and they provide amino acids and other functions as well, and they form these strange consortia. And so what we might be seeing now is a specialized, relatively recent in evolutionary terms, development of a new organelle, through bacterial capture, and to the point now where these things really require the cicada, and require, actually, in many cases, there's multiple different bacteria that require each other to complete these processes, which is just wild, kind of unbelievable that we're kind of seeing kind of relatively recent evolution of these types of, becoming part of the same biological system.
Yeah, so if you bring it to the human level, Fred, there's been a lot of talk around the human organism. I remember reading a paper a few years ago. It's a little far-fetched that there are nerve endings in the gut. The bacteria could actually utilize that to alter the behavior of the host, to get the right level of food into the system, and so on. So where do we stand on that general concept of human organism? And like you mentioned, a few pounds of the weight is actually just bacteria inside a human.
Yeah, so I think it's interesting. So some people have referred to this, and they've talked about like maybe the holobionts, this idea that maybe the unit of things that's evolving is not just a human, it's the human and all the microbes inside of it. And I guess that's like, and so with this actually this kind of consortia of like human cells and microbial cells, and we're all these interests aligned, and we're maybe a composite organism. That's cool. I like that idea. It's hard to have, but the evidence surrounding that I think is not great. In part, even in humans, the microbial community that we have on our skin or inside our gut is also very transitory. These things change quite quickly. They can change over our lifetime. They can change through diet and through even antibiotic use will change the microbial communities we have inside us. And so that kind of, the fact that it's so transitory, I think is maybe a knock against this holobiont theory. And also, it's not true that maybe microbes, they just want to they just, like humans, they grow and divide. And then, ones that are successful at growing and dividing are more represented in the next generation. And so their interests might lie elsewhere, right? And so, I don't know, I think... And then the effects they exert on us, yes, there have been studies that have kind of shown that maybe they might be important in things like, fascinatingly, like autism, mental health disorders, all kinds of potential things there. I mean, if that's true, that's crazy, right? Like, that's super cool. But it's really hard to show that, unequivocally, because there's so many factors involved in many of these diseases and issues that there might be a microbial component, but to what extent, I'm not sure.
Yeah, it's a very complex system. I saw some data that indicated that the human microbiome changes within a week. So, if you go from the US to, let's say, Japan, within a week, your microbiome is essentially different, replenished with different things. So this transitory effect that you talked about seems to indicate that it's the host itself sort of acquiring and assembling the right cohort of things, that is beneficial. I don't know, Sussane, if you go down to the amoeba level, what does that interaction tell us? It's probably not easier, but maybe cleaner to study what happens there.
Yeah, so I think it does serve as a very simple system where you could look at sort of mini microbiome formation. One thing that maybe, this amoeba system lacks is, even though it does have in my case, it has both a single cellular and multicellular life cycle. So you see some primitive sort of innate immunity going on there. It does lack more of a complex innate and, of course, an adaptive immunity, which I think is maybe one of the really interesting things about the microbiome in the mammalian system is what's going on with the immune system and those microbes. So I think some of the best sort of systems that have looked at that have been in those like germ-free mice systems. And so that probably serves as sort of the closest analogy to the human case, whereas in mine, I'm going farther back sort of to a more primitive state, an interaction system. But I think one thing I was just thinking about with what Fred was talking about and what you mentioned about the kind of idea about microbes influencing our sort of mental state, there have been some studies that show, for instance, that certain microbes can actually promote like serotonin production by the host, or even produce serotonin derivatives themselves. So I guess from that perspective, you could see how certain gut microbes could actually influence your mental state. So I think that's really interesting. But then going back to the immune system, one thing that I think is really challenging is how, as an animal, how it is that you learn how to counter the pathogenic microbes and destroy those, but not react to the beneficial or just neutral microbes like in your gut. And it seems like these beneficial microbes, or even just our general gut microbiome, is actually informing that response and tailoring that response as we develop.
And so, since amoeba is using bacteria as food, there is still resident bacteria in the system?
So in this case, not so much. I mean, certainly they're constantly encountering bacteria in the soil and in their environment. But in my case, if you think about the most of the time in their life cycle, so I guess I should back up and say the particular amoeba that I study is a social amoeba. So it goes from sort of a single cellular stage, which most amoeba exist in, to a multicellular stage that has sort of a differentiated structure, and then back to a single cellular stage. So if you think about things like a microbiome, you really kind of are thinking about a system where you have a multicellular organism, and microbes are maybe, existing sort of on structures and not just inside cells. And my system looks more at bacteria that are living inside cells rather than inside sort of multicellular structures. But I guess what I would say is that in this system, there are probably transient interactions at the multicellular stage that may be microbiome-like. But there are certainly some longer-term intracellular interactions that are occurring. But what effect they exactly have on the amoeba in the environment is not completely clear.
Okay. So Brett, termites are a lot bigger than amoeba, and so I expect we have more analogous. Sort of analogous to the human system, right? Interactions happening.
Yeah, to an extent. I think the gut of the termite is a pretty unique place just because it is basically a bioreactor based on the diet that the termite eats. But something I was thinking about that it maybe is a little bit more analogous to the mammal system when Sussane was talking about host-microbe interactions is, I think something that we haven't really talked about is the microbe-microbe interactions. And in a termite gut, we're talking about representative microbes from all three domains of life. They have protozoa, amoeba-like organisms. They have bacteria, they have archaea. They also have definitely viruses that are hanging out there. And that dynamic is also constantly changing. So you have the host and the food that they're eating, their immune system, and their physiology that's happening in the gut naturally. But all of these microbes are constantly interacting with each other. We've got intracellular symbionts in other symbionts and extracellular symbionts, and they're kind of all constantly interacting in this micro habitat. And so thinking about the immune system, I think something we haven't talked about is these antagonistic relationships between what we would consider are like beneficial microbes and these potential pathogens. And that's something that we're studying in our termite system. And there are some pretty exciting kinds of things coming out about, direct antagonistic interactions with specialized machinery for bacteria combat, and then of course things like secreted enzymes and antibiotics that can serve to protect not only the host, but this biofilm that these normal flora are all kind of working together to create and maintain.
Yeah, and I understand that in the case of the termite, the wood-based diet that they have, they require microbes to basically break down.
To break it down as efficiently as they do, the termite does have some endogenous cellulases. So there are cellulases in the termite genome, but a lot of the recycling of that wood is happening by microbes in the gut. Yes.
Okay. And so what's the lifespan of a termite?
So termites are actually incredibly long-lived. It depends on which caste you're talking about. So a queen termite can live for a decade or more. There's actually some
Oh, wow.
anecdotes out there that, particularly in Florida, where we have some invasive species of termites, that it's possible that those incipient colonies that came over in, wood crates or ships may have been there for as long as like 7 years before they rose to detectable levels. So one of those anecdotes in the pest community, find a termite, you probably have a million more, because it just takes so long. They're very stealthy, and they can live a long time. And a termite worker can go through several molts and stay as a termite worker. They're pretty long lived.
Okay. We'll take a quick break. When we come back, we'll talk more about all of these ideas. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. So we're back. Fred, Sussane, and Britt, we were talking about the microbiome, the interaction of the microbes with humans, but also other systems like termites, pests, even smaller systems like the amoeba. And in some cases, in the case of the amoeba, it's using microbes as a food source. In larger complex systems, there is a symbiotic relationship, maybe in the gut. But the picture is really complex. Some of them are beneficial, some of them may not be. The picture, the more you look into it, the more the picture getting a bit clouded. So Fred, I want to go back to sort of the history of this a little bit. One of the papers you had sent me, looking at the H. pylori bacteria, that there are different varieties of it, and we could potentially use sort of the movements of H. pylori to look at even the migration of humans around the earth, right? You want to talk a bit about that?
Yeah, I think that's one of the cool things about microbes. Is that they're small, and so their genomes are correspondingly small as well, especially bacterial genomes, which makes sequencing these genomes relatively easier than sequencing human genomes. And so I think as humans are quite interested about human population movements, how humans have evolved, how they've traveled throughout history. And that's quite hard to infer, right? So we can use archaeological information. That's been hugely important. We can even use human genome information to infer some kind of migrations and how populations have interacted. But fascinatingly, we can actually use microbial genomes, so bacterial genomes. And so studies have used H. pylori. Number one, because it's usually vertically transmitted. You often get it from your mother. It's transmitted within a community as well. So if you're living in the same household, you'd have similar strains of H. pylori. And bacteria, their genomes, as a rule, tend to evolve a bit quicker, or they change a bit faster. There's a number of reasons. It's mostly to do with kind of just their error checking mechanisms are not as efficient as ours. And, it doesn't really matter. But because they evolve quicker, we get more time resolution. So it's actually quite hard to infer human population movements just from our own genomes, because we evolve so much more slowly. But these kind of communicable bacteria that transmit between people have evidence in their genome about where they've been, where they are now, and where they might have been in the past. And so we can use these bacteria to create these kind of large-scale population movements that are with quite high temporal accuracy for the last 10,000 years, that often fascinatingly recapitulate things that we see in the archaeological record as well, of where people have been. So that's, I think, is just like a cool byproduct of having a microbiome, because the rates of evolution are different. They transmit, they tell you where you might have been and who you might have come into contact with. And so we could infer these types of large-scale migrations, which I think is just like a fascinating consequence of having a microbiome.
Yeah. The one of the papers I saw, so the evolution of the host microbiome as an ecosystem on a leash. So I just want to touch on this idea of this hollow genome idea. Is the host controlling it, or is the microbiome controlling the host? What is our latest understanding of that?
I think it's probably, I would argue, and I don't know, maybe Britt and Sussane will disagree with me, but I think there's probably some kind of tension there. In some cases, as a human body, you have an immune system, or any kind of maybe more complex organism that has an immune system. You can in part control what microbes are there, or you can at least provide the most beneficial environment for the microbes you want, and you can try to get rid of the ones that you don't want. And similarly, microbes will try to invade if they, we get lots of protection even in the human. So I know Britt mentioned that like the microbes in the termite guts can provide protection against pathogens or nasty bacteria or even viruses. Similarly in human guts. And I think one of the cool examples of this is that I guess we get to a roundabout way, but many pathogens actually can't invade when we have a healthy microbiome, and so they actually have to get rid of the bacterial flora, the bacteria we have naturally occurring in our bodies. And so there's lots of kind of cool examples. One of them is salmonella. So I'm sure most people have heard of salmonella. It causes diarrheal distress. It can be fatal if if you don't have access to like proper hydration therapy. But regardless, it's awful. Nobody wants like salmonella dysbiosis. It's terrible. Anyway, and the reason it does this actually, the reason you get terrible diarrhea is because it can't invade the gut on its own, because there's actually lots of other competitors there that prevent it from attaching, prevent it from growing. So what it does is it induces an immune response. So it'll actually invade into the gut lumen. Those cells will be destroyed by the immune system, and it will cause an inflammation. And this inflammation will kill off most of your resident bacteria. And then there's salmonella can hide. Some of the salmonella cells that don't invade will hide from the immune system and then recolonize now this brand new environment. Gone are the bacteria that were giving you some protection. And then it causes a horrendous infection that, results in, terrible outcome for the human for at least a few days. And it transmits to a new human. So it's, from termites to humans, potentially even to amoeba, these bacteria might provide some, our resident gut flora is really important, and many kind of pathogenic bacteria will have to try to circumvent that. In this case, salmonella is very clever and uses our own immune system against us.
Yeah, I mean, that's what's fascinating about this. Again, 4 billion years of evolution. Humans are really a sideshow if you think of the Earth, right? So, Sussane, amoeba hunting bacteria for food. That is a really complex system that is happening underneath our foot. And so do we see sort of the same sort of dynamic between the predator and prey relationship in there between amoeba and bacteria?
Yeah, so I guess when I think about that kind of dynamic, you see obviously, the predator is trying to phagocytose and actually digest the bacteria, and then there's selective pressure on bacteria to evade that, and there's many different sort of evolutionary strategies to do that. So one could be making just virulence factors or toxins that are secreted and sort of prevent an amoeba from even engulfing it in the first place, or even things like biofilm formation, to prevent being able to be sort of surrounded and engulfed. And those kinds of strategies are also important when you think about pathogens evading our own immune response. And then, of course, finally, in being able to be ingested but not digested. So if you can get inside of a eukaryotic cell, but then survive there and maybe capitalize on the nutrients and protection you get inside that cell, you could potentially evolve towards either a beneficial or pathogenic state. And I think what's nice about sort of this amoeba system is that those processes are also in play in mammalian systems with our innate immune cells like macrophages and neutrophils and dendritic cells, who are going around our body, looking for potential pathogens, whether they're bacterial, other microbial pathogens. They could be viruses as well. And then basically engulfing those through a phagocytic process and processing them, and then displaying the bits of those pathogens to inform the adaptive immune system. And so some of those interactions between basically evading that process or subverting that process in an amoeba could also be occurring in a mammalian system to basically select for bacterial pathogens, for instance.
Is there any sort of human therapeutic direction here?
Yeah, so I think understanding some of the basic virulence mechanisms of bacterial pathogens using a simple amoeba system could lead to, for one, just being able to counter those strategies, potentially through therapeutic interventions. The other thing I'm particularly interested in is understanding what are the amoeba factors that are leading to either more or less tolerance of potential intracellular invaders. And if we can identify some of those elements, maybe those could also be used as therapeutics. So one thing that that I'm kind of going into the direction of is looking at lysozymes, which are products that pretty much all eukaryotes make and are certainly important for mammalian systems that serve as sort of intrinsic defenses against bacteria. So we secrete them and they sort of enzymatically destroy other microbes. And amoebas do this too. And these lysozymes that are secreted, maybe you could consider harnessing those sorts of things as sort of factors to decontaminate bacteria, for instance.
Do we have anything practical today, or it's just a research direction?
That's a good question. I don't know if in the amoeba system, for sure, whether there's something that the amoebas are producing that is translated into a particular therapeutic, but I think we've definitely discovered more about the bacterial virulence factors that are contributing to pathogenesis. And that's more in systems where people are actually looking at human relevant bacterial pathogens. My interest is really in looking at natural interactions that are occurring between these amoebas and bacteria, and just beginning to sort of understand those natural interactions rather than use this system as a model for human relevant bacterial pathogens.
Okay. So Britt, so termites, like ants, have sort of a complex social system, right?
Yeah, they do.
And so what is you talk about lower termites and higher termites? So what exactly are those things?
So the names lower and higher termites refer to their evolutionary history. So lower termites are older lineages, higher termites are newer lineages, and really the dividing line is lower termites have protists in their guts and higher termites don't. So it's a human imposed bin that we put these termites into. But all termites do have these social interactions, and I think what's really interesting, kind of thinking about what Sussane was saying, what we can learn from these systems, termites have hygiene, and they clean their nests, they clean each other, they will actively groom things like pathogenic fungal spores off of each other, and they also do something that I know Sussane and I have talked about. They basically do the equivalent of a fecal transplant on each other after molts or after dysbiotic events. So when their microbes have been disrupted because of a molt or an infection, they will feed each other poop and refaunate their guts because, as we've talked about, without a healthy microbiota, they aren't healthy as a whole. And so I think that's something interesting that we can take away from termites, and we are implementing in human systems. You know, there are some FDA-approved fecal transplant as a therapy.
And so they're community living, the termites community living. that is also, I would imagine, influenced by the bacteria, right? So, what kind of effects do they have in some terms of, I'm thinking about ants. Do they affect organization structure, where they are found, and so on?
So there's some thought about termite evolution being really driven by this reliance on gut microbiota. So social living may be a product of this reliance on gut microbiota. In terms of how their casts are regulated in termites, the mechanisms are a little bit different than in things like bees and ants.
Yeah.
It's hormonally driven, and we've talked about how bacteria may play a role in host hormone production and things like biogenic means like serotonin. We haven't made that link explicitly in termites, but there seems to be some evidence that reformation of termites after molts can really change the trajectory of their life history. So we don't have a direct mechanism in relationship to cast, but we do think that these gut microbiota have a really big impact on termite evolutionary history in that transition from cockroaches, which are kind of semi-social and a little bit gregarious, to termites, which are truly social and interreliant on each other.
Yeah, and I think there's other interesting examples from the insect world, like using their microbiomes for detection. So it seems like bees, for example, might be detecting. So how does the bee know whether it's in its right nest or not? Well, often it can remember where it's been flying, but how do then bees also recognize themselves? And so there's kind of lots of pheromone communication. And there's been some experimental evidence to suggest that these kind of pheromones are directly linked to the microbiome that these bees carry. So these bacteria are altering their the pheromone composition, and allows bees to recognize I'm from this nest, or this is an interloper not from this nest, and that's directly being driven by a microbial community. But you do also raise an interesting point there, Gil, about the fact this transition from sociality, from kind of just living on your own to living in communities. It's a huge question. Like, how do social environments cope with diseases? Because you're more prone to spreading diseases when you live in kind of a colony. You live together in a city. We've seen this happening, even for humans. This happens when you get lots of people close together. Diseases spread much more quickly, and so there have to be you get a lot of benefits from maybe living kind of social life. Division of labor, for example. You have things that specialize on getting food, or things that specialize at reproducing, but you also get some kind of downsides. And one of the major downsides is the spread of disease. So maybe you also have to evolve then more complex immune systems or behaviors like hygiene, cleaning, that can counteract these downsides of being social.
Yeah. Talk about social and close interactions and COVID-19. So the other thing I want to touch on, Fred, is obviously antibiotic resistance has been a problem. Because of economic and other reasons, there hasn't been a lot of research in this area. Commercial research, I should say, in pharmaceutical companies. And so we are sort of pushed against the wall now in terms of antibiotics. And so where do we stand on that now? The current understanding of bacteria, is it going to help us design more effective antibiotics? Or is it not the direction we want to head? Are there different ways to handle bacterial infections to humans?
Yeah, I think that's an interesting question. So we know that our microbiome is so important for many, many things, and it might be involved in, things that sound far-fetched, but may be true from mental health to just, normal, everyday, gut function. So, but the problem is when we, try to kill these pathogens, we use we use a sledgehammer, essentially. We use an antibiotic, and antibiotics tend to kill all bacteria. That's why they're effective. They work really well, but they're not very targeted. So that's an issue. Moreover, and also, when you target all bacteria, then the chance for resistance genes to emerge and spread are just greatly increased. And so there's a lot of thought about how can we tailor. We live in an age of diagnostics, of incredible diagnostics. So often we can, hopefully in the future, we'll be able to quickly figure out what's wrong in terms of like bacterial pathogenesis. We could identify it pathogen quite quickly, and it'd be nice to have a treatment then that could target that. How we make a treatment, that's less clear. So, of course, maybe there's some chemicals that tend to target certain bacteria over others. We know that actually, but using bacteria themselves might be a good option, because bacteria, it's a competitive world out there for a microbe. They've evolved to kill each other very effectively. In fact, most of our antibiotics come from streptomycetes that produce broad classes of these types of antibiotics. But even more so, they produce peptides that are quite targeted at kind of competitors. And fascinatingly, an idea that's coming back into fashion is using viruses. So just like humans, bacteria have their own viruses. We often call them bacteriophage. And so this was an idea, and this is not a new idea, using viruses to treat bacterial infections. This has been around for almost 100 years now, I suppose. But back in the early 1900s, it was discovered that you could get viruses that could kill bacteria. This is kind of pioneered by Felix Derrel and Georgi Eliva, who was the Georgian, but the Republic of Georgia doctor there. And they used sort of viral concoctions they got from the sewers to treat quite effectively bacterial diseases. And so there is some work now trying to use specific viruses in the case of like highly antibiotic resistant infections to kill these bacteria with some success. And I know this is probably something, I think Sussane can speak a bit more about, because she in fact worked at the Georgi Eliva Institute in Georgia. I think it's in Tbilisi. I don't know where it is, but
Tbilisi.
On looking at some of these kind of viral treatments for bacterial diseases.
Yeah, Dan, you want to talk a bit about that?
Yeah, sure. So, and I think one thing Fred brought up that was really important is this specificity. So, one thing that's been really why antibiotics became so popular was that it was that sledgehammer approach, a silver bullet that basically if you had almost any type of bacterial infection, you could just take this pill and get rid of it. You didn't really need to know or identify what the particular bacteria was. Of course, that means that you also got rid of all of the potential beneficial bacteria that you had as well, and that was a potential downside. But now with this idea of being more precise at being able to detect exactly what potential bacterial species you're infected with and is causing the problems, that allows us to tailor an approach to combat it. And this is sort of the benefit now of this idea of phage therapy, where you're taking maybe a collection of viruses that are targeting very specific species or even strains of the bacteria. And these are sort of thought as also self-replicating therapies, because as a virus, as soon as they infect a cell, they're going to reproduce thousands or hundreds or thousands of progeny, which can go on to continue to target that selectively that bacterial pathogen. And so this is definitely something people are coming back to. And I think, as Fred said, there's been sort of mixed success. And I think we're seeing it more maybe being used more now in cases where people have antibacterial resistant infections that we just haven't been able to treat the normal way. This is kind of the last ditch effort and has been really successful in those cases. But there's a lot of other things that make it challenging, like how do you, first of all, identify that pathogen, be able to isolate a bacterial virus that targets it, and then deliver it to the appropriate area that's infected, and that might be internal to someone. But I think it is really exciting. And then with newer techniques to actually genetically manipulate these bacteriophages or change them in some ways to make them more effective is potentially on the horizon.
Yeah. So is it possible, Sussane, to conduct experiments to essentially evolve bacteria? So given this predator-prey relationship, would we actually control those mechanisms to essentially create newer forms that might be beneficial in some way?
Yeah, and maybe Fred can even talk about that a little bit more. But bacteria are, evolve quite rapidly because of their generation times. And so I guess if you were to supply the correct type of selection pressure, you could evolve more beneficial a microbiome bacteria, for instance, or in this case, if we were talking about bacteriophages, you could, evolve bacteriophages that target certain types of bacteria or even bacteria that are specifically resistant to antibiotics and then just target those. But I think if you're applying the right kind of selection pressure, there's certainly a way to potentially evolve the organism that you want, although I think evolution is complex and it's hard to necessarily control the trajectories. But that's definitely something I think Fred could probably elaborate a lot more on.
Yeah, I mean, I think there's probably two avenues here. I mean, the first is we might already be doing this anecdotally. If you have lots of cultures eat a lot of fermented food, which is chock-a-block full of microbes that often are very beneficial. There's been some evidence suggesting that if you are on an antibiotic treatment, eating something like yogurt is quite good for you because it reseeds your kind of microbial community with lactobacillus, which can have positive effects. And we've been eating yogurt for, thousands, tens of thousands of years. So these are kind of, not only does it save our food, but it might also protect our guts. But yeah, there's some thought that maybe one of the things we can do if we don't have effective treatments for killing microbes is, actually inserting a microbe into our body that can outcompete the pathogen, or a version of the pathogen that isn't as good, but grows a bit faster. So a lot of the mechanisms that make us sick, these virulence factors that will interact with our body and cause the kind of bad outcomes that cause the disease, they're incredibly metabolically expensive to produce. It takes a lot of energy for microbes to produce these. And so if we engineer microbes that don't have them and then seed these microbes into the human body, they'll actually be able to outcompete or grow faster than many of their kind of pathogenic competitors. And then they can actually cause, through this competition, they don't grow as well, and then the immune system can come in and do its job. So kind of like a Trojan horse style, where you kind of genetically engineer the bacteria so it can compete with the disease-causing strain, but then allowing time for the immune system to clear the infection. So this has been suggested. There's been some work done. I mean, so it's hard to get approval to do this in humans because you're putting in a microbe that might evolve to also be pathogenic. But there's some example in agriculture where people have taken these strains. There's a lot of kind of cool new agricultural biotech startups who actually do this, engineer the bacteria to outcompete the blight-causing, disease-causing strain to actually relatively high success.
Mm-hmm. So in conclusion, Brittany, I know that you got a NSF grant recently. This is looking at again gut microbiome in pests like grasshoppers. Right? You want to talk a bit about that and what your sort of your future direction of research might be?
Yeah. So this NSF grant is an institute grant with several other institutions, and my prong of the project is to describe the gut microbiota of Schistocerca locusts. So these are a genus of locusts and short-horned grasshoppers that have phenotypic and behavioral plasticity. So they do things that we might typically think of as like grasshopper-like, and then they also do these like biblical plague style, just swarms and migrations and complete defoliation of landscapes. And so what I'm looking to do is identify the microbes that are in their gut. We know that they have some, but we don't have a great handle on what it looks like across species, and particularly when they do this shift, this massive physiological and behavioral shift, what is happening in their guts. And so the hypothesis being that there could be a microbe in there that is playing puppet master, maybe changing some of the circulating hormones or things like that, that could be modulating their behavior or changing how they digest their food. And so that's kind of what we're looking at, trying to find a core microbiome in this genus, but also seeing if there are any key players that seem to predict outcomes for the host's physiology and trajectory.
Right. And Sussane, again, in conclusion, what is your sort of future research direction? I know you're working on some bacteria-host interactions, and maybe from that, learning how complex microbiome effects could be.
Yeah, and one thing actually, since we mentioned the bacteriophages, another thing that I've been integrating into my system is to basically find these bacterial viruses that infect the particular bacterial symbiont that I study that infects these amoeba. So you've got this 3-tier system of now bacterial virus that targets the bacterial infection, I guess I would say, of the amoeba host, and then adding all of those 3 components into the system and asking how they interact with each other and influence each other's interaction and dynamics and evolutionary trajectories. So it's kind of like a phage therapy-like approach, but to see what the long-term evolutionary trajectory of those interactions are within the system.
And Fred, again, in conclusion, what is your future research direction? It seems like using bacteria against bacteria seems pretty profitable.
Yeah, so that's definitely an interest. So I'm very interested in the kind of toxins bacteria produce and how that affects their competition and how that can affect diseases in like model systems that we use in the lab. And we also have an arm of research trying to understand how, as phage therapy becomes more popular, we might want to know about how viruses evolve a bit more. Because, there's people suggesting using cocktails and how that phages, different types of phages, and how they interact with each other, I think is going to be very important in understanding how effective some of these treatments are going to be. And I suppose just as a final thought, one of my colleagues here at the University of Missouri, St. Louis, he works on salmonella, and one of the interesting things coming out of his lab suggests that one of the most important parts of its infection cycle is being able to attach to the gut lumen. And if it can attach and it can invade and it can induce this immune response, then you're going to get some kind of salmonella infection. But turns out the salicylic acid is pretty potent at preventing this from happening. So if you're out traveling to a foreign country, you're worried about foods, how good the food's going to be, an aspirin a day is potentially not just anecdotally correct, it might be scientifically the correct thing to be doing.
Yeah, I definitely need that, Fred. This has been great, guys. Thanks so much for doing this on a weekend.
Yeah, thanks for having us.
Yeah, my pleasure.
Yeah, thank you.
Have a great holiday.
You too.
You too.
You too.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.